Hey guys, so last week I went over action potential in the neuron. To this week, I'm just gonna go over the muscle structure and the neuromuscular junction, as well as the cross bridge phase, actin and myosin. So first off, I'll start off with the characteristics and functions of muscle. And here we have four characteristics of muscle. One being the excitability, meaning that your nervous system is able, or your muscles are able to react to certain things, is responsive. You're able to run. Two, contractility, ability to shorten. That would be an example of that would be like the cross bridge formation, the way your muscles, they have the ability to contract or shorten. And three is extensibility, the ability to stretch, and elasticity, the ability to recoil. So here are some functions, or the 8 functions of muscle. Produce movement, maintain structure or your posture, stabilizes your joints, generates heat as they contract, respiration, communication, constriction of organs and vessels, and contraction of the heart. So there's three types of muscle tissues. The three types of muscle tissues is skeletal muscle, cardiac muscle, and smooth muscle. So remember, in lab, we have to differentiate between the three. So here's some differences between the three. So skeletal muscle attaches to the skin and bones, and that's going to have striations here. And then we have, it's also voluntary, meaning that you consciously move your muscles. They're not involuntary, meaning that they don't just happen by themselves. The function of that would be body movement. So we are able to run, walk to our own ability. And then we have cardiac muscle. So cardiac muscle or cardiac tissue is only found in the heart, nowhere else in the body, and that is also striated. This is a cardiac muscle. You can see the striations. This is involuntary, so we do not have to think about pumping blood or keeping our heart pumping. So that means that is involuntary. We have smooth muscle. So this is found in the walls of hollow organs like our stomach, part of our urinary tracts. This has no striations. So if we look at number 2, this is smooth muscle. This has no striations. And this is also involuntary, so we don't have to think about using the bathroom or to urinate, or our digestive tract. We don't have to think how to do that. We just go ahead and do it. So it's involuntary. And here, I just want to specify on cardiac muscle, we have these cell-to-cell attachments, particularly in cardiac muscle. So each cell in the cardiac muscle is attached by this special cell-to-cell attachment called intercalated discs. So you can kind of see it in this picture, these lines. These are called intercalated discs. They join each cardiac cell together. Okay, so now I'm gonna break down the muscle. So skeletal muscle has three major features. So it's supplied with nerve fibers and blood supply. So it has nerve arteries and veins. It has it's surrounded by connective tissue sheaths. So we have the epimysium, the perimysium, and endomysium, and attachments. For example, for attachments would be like the origins and the insertion that we're doing in lab. Origin meaning that it's attached to an immovable bone, and the insertion is attached to a movable bone. And we also have indirect and direct. So here's an example of direct attachment. So if we look at the temporalis, we have a direct attachment here where it attaches to the aponeurosis. This part is the aponeurosis. So if we go back, so direct means that it's the connective tissue wraps extends beyond the muscle as rope-like tendon or sheet-like, just like the aponeurosis. And indirect would be like the epimysium, how it's all fused into one. So it turns into the epimysium, and then it turns once it gets down to the bone, it's fused to the periosteum of the bone, and then it gets down to the cartilage, and then that turns into perichondrium. So here's a picture of the indirect attachments. Okay, so we have the layers of the muscle. So starting off with the entire muscle organ. This is the entire muscle organ. The entire muscle organ is surrounded by the epimysium. And then we get down to the fascicles. And those are going to be surrounded by the perimysium. Here, let me see if I can. Yep. Okay, here we go. So I'm going to draw it out and break it down. So this is the entire muscle organ. And we have veins. And what's surrounding the muscle organ is the epimysium. And then I'm going to exaggerate this a little bit, but these are the fascicles. So I'm just going to draw this huge fascicle here. So this is the fascicle. And this is going to be surrounded by the periosteum or perimysium. And inside the fascicles, we have muscle fibers. So again, I'm gonna exaggerate this. And here's the muscle fiber. And the muscle fiber is going to be surrounded by the endomysium. And inside the muscle fiber, we're going to have the myofibrils. And another note is that underneath the endomysium is the sarcolemma. So right on top of the sarcolemma is the endomysium. So the blue is the sarcolemma. And the difference between the endomysium and the sarcolemma is that the endomysium is a connective tissue, and the sarcolemma is a membrane. So it's gonna have the phospholipids, and it has permeability. It has all the ion channels on it. Sorry. Let me erase. Oh, excuse me. And then here inside the again, this is the myofibril. And in here, we're gonna have the myofilaments. And that's gonna be the actin and myosin. And somewhere around here, you're gonna have a T tubule. I'll get to in a second. I'll get to what they do. And then you have the SR right here. So this is the drawing of a broken down skeletal muscle. So again, you have the muscle organ surrounded by the epimysium, and then you have the fascicles inside the entire muscle organ, and that's going to be surrounded by the perimysium. And then we have muscle fibers inside the fascicle. That's going to be surrounded by the endomysium. And underneath the endomysium, we have the sarcolemma. Inside the muscle fiber, we have myofibrils. And those myofibrils are surrounded by the sarcolemma. And inside the myofibrils, we have the myofilaments, the actin and the myosin. So this is a visualization of what I was just describing. You can check that out. And so I'm going to get into the muscle fiber. So the anatomy of the muscle fiber. So like I said, we have the sarcolemma. It's a plasma membrane that surrounds the muscle fiber. Which is, like I said, is underneath the endomysium. And we have sarcoplasma that's in the that surrounds the muscle fiber and I'm sorry, the inside the mixed in with the myofibrils and the sarcolemma. And that the sarcolemma is going to have it's going to consist of glycosomes and myoglobin. Glycosomes, what they do is they store glycogen, and myoglobin, they store oxygen. So I'm pretty sure that most likely will be on the test. And we have, I'm sorry, the muscle fibers has modified organelles, the myofibrils, and it has the sarcoplasmic reticulum and T tubules. The sarcoplasmic reticulum is going to surround the myofibrils, and its function is to regulate the calcium level. So it stores and it also releases calcium. And then we have T tubules. And what T tubules do is they allow electrical transmission to reach deep down into the muscle fiber and it reaches the myofibrils, and that allows or that connects to the sarcoplasmic reticulum, and from then on it allows calcium to be released into this sarcoplasm. So this is just what I was trying to tell you earlier, the difference between my endomysium versus the sarcolemma. So structure-wise, the endomysium is a connective tissue, and the sarcolemma it has a phospholipid bilayer. So if you guys remember, the phospholipid bilayer is the where you have a phosphate head, and then you have the lipid tails. And that's going to be the entire membrane. So the triad, we've mentioned, the professor has mentioned this a couple of times in class, but what it consists of is the sarcoplasmic reticulum. It has these terminal cisterns, or cisternae for plural, and that's where the calcium is released, and the T-tubule, you see right here, going down, that's the action potential is going to go down there, and in doing so, it's going to activate these ion channels in the sarcoplasmic reticulum, or specifically the terminal cisterns right here. These are called the terminal cisterns. And that's going to release calcium. So myofibrils have 4 features. The striations that we see are actually the Z discs and the myofilaments. All of those striations that we see in lab, that's what those are. And it has sarcomeres. Sarcomeres, if we're talking about the Z discs and the I believe it was the M line. So this entire thing right here, the Z discs, or Z line, you can also call it the Z line. This is what makes a sarcolemma. So the Z lines is just the boundary of each sarcomere. So this is one sarcomere right here. And it's also going to consist of myofilaments. Those myofilaments are actin myofilament and myosin myofilament. Myosin, you can call it thick filaments, and actin they call thin filaments. And the myosin filament, that's where you have these myosin heads. And those are what going to link up to the actin filament in order for a contraction to happen. I'll get to the slides and the whole, all of the steps and what happened in a bit. And we also have these other proteins called tropomyosin and troponin. Again, I'll get to those in a second. So here's a picture of the contraction happening. Okay, so here's troponin and tropomyosin. So we have tropomyosin. What it is a strand that wraps around the actin filament here. And when calcium is not connected to the troponin, the tropomyosin covers the active sites on the actin. Let me see if I could. Okay, there we go. So I'm going to draw an actin filament here. So actin is going to have these active sites where myosin heads are able to bind or link. So this is the active sites. And then we have troponin. I'm sorry, tropomyosin. So tropomyosin is going to be this long strand that wraps around the that actually it covers the active sites. So it covers the active sites on actin. And then we have troponin. It's on the on the tropomyosin. And what this does is when it's binded to a calcium, so when binded with um, with calcium, this protein troponin is able to change shape when it's binded to calcium. So when binded to calcium, it changes shape. And in the act of it changing shape, it moves the tropomyosin, exposing the active sites on the actin filament. So when it exposes, the moment that it exposes those active sites, that's when the myosin is able to attach or link up to the active sites on the actin in order for contraction to happen. So we have a myosin strand here. So these are going to be the myosin heads. Okay. So the 4 steps in the muscle contraction, I'll get to that in a bit. I think there's some more stuff that I want to get to first, but this is just like the basics, understanding of what actin is and what myosin is and what actin has, or what it consists of. So like I said, it has tropomyosin, which is a long strand, and it covers the active sites on, it could either cover or expose, I should say, can expose binding sites. So it has the ability to cover or expose those binding sites on actin. And troponin, when it binds to calcium, that's when it changes shape and it moves the tropomyosin strand, which exposes the active sites on the actin. And by doing that, the myosin heads are able to link up to those active sites or those binding sites on the actin. Okay, so muscle contraction. What is muscle contraction? Um, So like I mentioned, muscle contraction is the activation of the cross bridges. So the moment that the triple myosin strand, we see here it's in orange. Once those active sites are exposed and the myosin head is able to link up to those active sites, it creates a cross bridge. So this whole formation here is called a cross bridge formation. And when they detach, that's when contraction ends. That's it's no longer contracting. So this picture is just a picture of a of contraction happening. All right, so this is leading up to contraction. We have three major events that happen before we get to the cross bridge cycling of the where the myosin and the actin filaments attach. Leading up to that, we have three other events. We have the neuromuscular junction. This is where the motor neuron and the muscle meets, or specifically motor neuron and the sarcolemma. Two, we have the muscle fiber excitation right here. So the moment the neurotransmitter is binded to the neurotransmitter receptors, or the chemically gated sodium channels, once they open, that's usually the second the start of the second step, muscle fiber excitation. And then the action potential continues to go down. Down, and this is where excitation contraction coupling has or begins. So the release of calcium. And I'll go in depth in a bit. So starting with the neuromuscular junction. So here is the 6 major steps. So first, action potential arrives at the axon terminal. The voltage gated calcium channels open, allowing more calcium to enter the neuron or the axon terminal. The entry of calcium allows the synaptic vesicles to release the neurotransmitters. Transmitters eventually bind to the neurotransmitter receptors, or the chemically gated sodium channels on the sarcolemma. And those open up, letting more sodium in. And that sodium is gonna increase in concentration, allowing more sodium channels to open. So let me draw a picture. So leading up to muscle contraction. first step is the neuromuscular junction. Okay. And here we have the axon terminal. So this is the sarcolemma. And then we have the axon terminal. Okay, so the first step is that we have action potential arriving at the neuron or the axon terminal. If you're not too familiar about what happens or how action potential is generated in the beginning, I did a session on that last week and it's up on the YouTube channel, so you can check that out if you still don't understand how that happens. Would recommend checking that out. So first step is action potential arrives at axon terminal. And then we have these synaptic vesicles. These are going to be carrying the neurotransmitters. These are, and their specific name is actually acetylcholine. So these are the neurotransmitters. You can call them ACH, or not too sure if I'm spelling it right. Acetylcholine. Acetyl. I think I spelled that right, but I think you get the picture. And then this is the action potential. These are the synaptic vesicles carrying neurotransmitters. So the information or the action potential, you can also call that information because it's also being, the information is being transmitted, or the action potential is being transmitted. So this would be pre-synapse, meaning it's in the act of sending information. So this is sending info. So continuing. The brighter color. And this right here is going to be the voltage gated calcium channels. Voltage gated calcium channels. So step 2 is that when action potential goes down to the axon terminal, it opens up these calcium channels. So calcium ion, or specifically voltage gated voltage gated calcium channels open. So you have calcium here, and start going inside. Three, you have so you start getting a large concentration or high concentration of calcium, and that's going to allow the release of the neurotransmitters. So calcium causes synaptic vesicles to release neurotransmitters, or ACH. So here's a picture of the synaptic vesicle letting out the neurotransmitters. And at the sarcolemma, so we're going to have these chemically gated channels. So this is going to be chemically gated channels. Specifically, they're chemically gated sodium channels because they're these are going to let in sodium into the chemically gated sodium channels. So step 4. So you're gonna have the neurotransmitters are going to bind to the chemically gated sodium channels, and that is going to allow the entry of sodium to go inside the membrane or go through the membrane. So I'm gonna call it as ACH for short. So this is a neurotransmitter. So ACH binds to chemically gated sodium channel. Allowing sodium to flow through sarcolemma. So let me draw some sodium ions. So I'm gonna flow through in here. And here at this near the junction, you're gonna have more voltage gated sodium channels. And so like last week, like I said, I was talking about what depolarization is. So just briefly, depolarization, the cause of depolarization is when a large concentration of sodium builds up and it starts creating, I guess the membrane becomes less negative. So if we look, this membrane is has a normal polarity, right? But once depolarization happens down here, it's going to flip the charges. So this is going to be negative and this is positive. So it's gonna make the membrane less negative. And by doing so, it's going to open up more sodium channels because of the less negative membrane. So here, the moment, I guess, though this is the way I see it. The moment that the neurotransmitter binds to the chemically gated sodium channel, and once it starts letting in sodium into the sarcolemma, this stage is going to be called the muscle excitation. So let me go back to the slides. So this is what we just did. So again, we have action potential that arrives at the axon terminal. Voltage gated channels open, allowing more calcium to go in. Calcium entry causes the release of neurotransmitters, and the neurotransmitters bind to the chemically gated sodium channels, and that is going to allow sodium inside the sarcolemma. And the next step after that, so okay, so here's like what I just diagrammed, or what I just drew. And moving on to muscle fiber excitation. The depolarization, or the action potential, is going to go down the sarcolemma. So I'll call this step 6. So action potential, or depolarization, goes down sarcolemma. 7, I would say depolarization of of the sarcolemma causes voltage gated sodium channels to open. So more sodium flows in. And now here, let me erase this really quick. This is the T tubule. And when the depolarization or the action potential goes down the T tubule, that step is going to be called the excitation, contraction, coupling. Trying to figure out where to write this. Okay, I'll just write it right here. So step 3, we have excitation, contraction, coupling. So I'm gonna draw the SR or the terminal cisterns right here. And the depolarization of the going down the T tubule, and that's going to allow more sodium channels to open, and sodium starts to flow in through, and that's also going to cause more calcium ion channels to open. So this is going to release sodium channels. I'm sorry, calcium. So 7, so 8, I would say. Here, let me highlight these steps, that way it doesn't look too confusing. So the highlighted is the major steps. So again, this isn't contraction just yet. We'll get to there. It's the last step, it's the 4th step. This is all leading up to contraction. So depolarization goes down T tubule. In doing so, it opens up voltage gated sodium channels. And the sarcoplasmic reticulum releases calcium. Or I should say the calcium channels open and then release sarcoplasmic reticulum, calcium ion channels open, and calcium begins to flow through. So once these calciums start flowing around, this is going to lead to the last step in cross bridge cycling. And these are going to bind to the troponin. So let me see if I have. This is the fourth step, or the very last event. This is a cross bridge formation. So you have the actin, and you have the active sites on the actin. And then you have the tropomyosin covering the actin, the active sites. And then you have the troponin. And then you have the myosin. Yeah, the myosin heads. So this calcium is going to come over here and it's going to bind to the troponin. And it's going to move It's going to move the triple myosin head or the triple myosin strand. And once it moves the triple myosin strand, it exposes the active sites on actin. And those active sites are going to let the myosin heads to attach. And this whole step right here, this is where muscle contraction happens. So I have a whole slide in what happens in that last the muscle contraction. So let me get to that. So like I said, it's called the cross-bridge cycling. So when the muscle is relaxed, that means that no calcium is attached to the troponin, and since there's no calcium attached to the troponin, it doesn't move the triple myosin strand that would allow the myosin heads to attach. So here, there's 4 steps that happen in that cross-bridge cycling. So I'm just gonna copy this over and okay. So this is cool. So this is where muscle contraction occurs. This is called the cross bridge cycling. So in step 1, we have the cross bridge. Oh, I'm sorry. Before I get to the steps, I want to talk about the ATP, ADP, and the phosphate. So gray is going to be a ATP, purple is going to be ADP, and the orange is going to be the phosphate. So first, what happens? the myosin head needs to have energy in order for it to slide the active filament for contraction. And how it gets that energy is by the ATP binds to the myosin head right here. So you see the ATP is coming to the myosin head, and over time it undergoes this process called hydrolyzation. The professor said we don't need to get in depth in what that is, but just know during hydrolyzation, it converts to a phosphate, an inorganic phosphate, and ADP. So you have the ATP and it's going to bind to the myosin head. And in that process, it hydrolyzes to the phosphate and the ADP. It generates this energy, and it puts the myosin head in what the professor called a cocked position. So this is going to move the myosin head back. And in the process of hydrolyzation, it's going to convert the ATP into ADP and the phosphate. So this is phosphate, inorganic phosphate, and this is the ADP. So ATP over hydrolyzation, it makes inorganic phosphate and ADP. And the position that the myosin head is in, this is called the cocked position. So the energy dispersed energy from the hydrolyzation of ATP to phosphate and ADP um, puts the myosin head in a cocked position. So another way to think about this is that if you think about a gun, you cock it back, and you have the when you pull the trigger, it releases this energy, right? So that energy, when you pull the trigger, that's the motion um, that you can think of the myosin head. So it's cocked back, and now it's ready for it to connect to the actin filament, and then it's going to push it in order for contraction to happen. So this, in other words, you can call this high energy, or you can also think about it as a slingshot. So when you have the slingshot, you have the you pull it back, and and you have, once you let it go, you have that tension when you pull the rubber band back, and you have that tension, and once you let it go, it's gonna let whatever you have on there out. So here's the first step. So the first step is the cross bridge formation. So you have the triple myosin is going to connect, or is going to expose the active sites on the actin. So just imagine that the triple myosin is moving, and it exposes the active sites. So you have a calcium that comes in here, and it's gonna bind to the troponin, and the troponin is going to change shape, and the troponin is going to move the myosin, the triple myosin. So when you have the myosin head in this high energy state, it's going to link up, it's going to connect. So this is the process of it connecting to the active site. So here, this is where high energy myosin head attaches to actin. And here you still have the ADP phosphate connected. Let me refer to my notes really quick. So here, so the myosin heads connect to the actin, and this, when it connects to the active site, that's what they call a cross bridge. So when you have the so here, so just imagine I just zoomed up here. This is the actin, and this is the myosin. So this is called the cross bridge formation. So it's in the act of contraction. So then in this step, you're also gonna have the phosphate is going to detach from the myosin head, and in doing so, that's going to create a stronger bond between the myosin and the actin. And then second, you have the power stroke. So the power stroke, meaning that it's that's when the contraction is happening. So it's moving the actin. So 2 is the power stroke. So here, the actin, I'm sorry, the myosin is going to pull the actin filament this way. So here you have the myosin head pulls actin toward the M line. So, and also in this stage, the ADP is going to be released. So once the ADP is released, that's going to cause the that's what causes the sliding of the actin and the myosin. So if you can see here, the movement here, and then the ADP releases, and the myosin head moves the actin. And then we have the cross bridge detachment. So this is the process in which the myosin head detaches from the actin. And what causes the detachment of the actin is the ADP. I'm sorry, ATP. So the ATP you're gonna have another ATP molecule come in, and it's going to attach to the myosin head. And when that once the moment that ATP attaches to the myosin head, that's what detaches or causes the detachment of myosin and actin. And then fourth and last step of the cross bridge formation is the reactivation of the myosin head. And you can think about this step as like reloading. That's the way I think about it. It's being reactivated into that high energy state. So it started the moment that it detaches, it's at a low energy state. So it has no energy. And once it attaches to the ATP, again, you're gonna have the hydrolyzation of ATP to ADP and phosphate. And that energy from the hydrolyzation of ATP is going to cause the myosin head to be pushed back right here. So it gets pushed back, and that's going to cause the energy to be dispersed to the myosin head. And then it's a whole cycle. So this continues over and over and over again during contraction. So this is an overview. So you have, first you have the cross bridge formation right here, and you have the working power stroke. So the ADP and the phosphate releases, and you have an ATP molecule that comes in the third step, and it detaches the myosin head. And then fourth and last, you have the cocking of myosin head. So ATP hydrolyzes to ADP and the phosphate, and it's a recurring cycle. It continues. All right, so coming up to the last couple of sections, we have muscle twitch. So muscle twitch is a simple contraction resulting from a muscle fiber response to a signal action potential from a motor neuron. And when muscle contracts, it also produces tension. So like if you have a 10-pound, I don't know, just imagine this is a muscle, and then you have another 10-pound bell, and it's being pulled on both sides. This is going to create tension. So that tension, like when you lift weights, it's creating tension. It's contracting, and it also produces tension. So a motor unit consists of two components. One, you have the motor neuron, and two, you have the muscle fiber. So the way I think of it is motor unit equals motor neuron plus neuron fiber. So you have the cell body here, and you have the neuron, and then you have the muscle fiber. So all of that equals one entire motor unit. And the three phases of muscle twitch, one is the latent period, second is the period of contraction, and three is the period of relaxation. So one, the latent period is that's going to refer to the muscle excitation coupling. So remember, we were talking about how once the action potential reaches the terminal, it goes down to the neuromuscular junction, and then you have the action potential goes down the T tubules, and then the calcium is released in the sarcoplasm. So that's latent period is going to consist the events of the excitation contraction coupling. And then two, you have the period of contraction, and this is going to refer to the cross bridge formation. And this is the myosin head connects to the actin filament. And then you have three, the period of relaxation. This is just where calcium begins to re-enter into the SR, or the sarcoplasmic reticulum. Okay, and lastly, we have rigor mortis. What this is so during those 4 steps of cross-bridge cycle, the calcium comes in. Oh, well, first off, this happens when somebody has died. So a couple hours after death, during the cross-bridge cycle, there is no ATP that's able to detach the myosin head from the actin filament. So this doesn't happen, and this doesn't happen. So in other words, it just attaches and it stays that way. It stays attached. So the calcium is going to continue to build up, and your muscles are going to stay contracted, and that's what stiffens up the body after a couple hours of being dead. And that pretty much sums up the lecture. So I'm just gonna have a quiz